The gene revolution, I'm sure you are a very educated crowd and you know what I'm talking about here. But as a scientist, I was a soil biologist, as Anne just explained. I was a plain soil biologist. I couldn't even spell DNA like most people. And about 25 years ago, I realized that funding was really short in soil biology, and all the money was in biotech. And so I did what any normal person would do. I just learned DNA, and I learned biotechnology, and I learned genetic engineering, and I became a genetic engineer, and there was plenty of money. And it's the same situation today. So there is no doubt that the gene revolution has been incredibly successful, and most people don't really know what it is about. Many people are concerned. Many people know that it's not good, or maybe perhaps it's even dangerous or toxic, but they don't really know why or how. So I'm going to spend a few minutes really making it really simple. And when you leave the room, you will understand and you will really know what it's about. I'm going to talk about what our GMOs are, and I'm going to make it really very simple. And then I'm going to talk about really what they deliver in the field, because there's a lot of hype. And then I'm going to talk about the pesticide that goes with GMOs, that goes with genetic engineering technology, because that's what it's about. It's about selling a pesticide. The whole gene revolution is about selling one chemical. So what are GMOs? What GMOs are, 90% of all engineered plants on the planet today are engineered to resist, to tolerate one chemical, the herbicide Roundup. So everything else that you hear about, like golden rice, or even BT, or drought resistance, or salt tolerance, or all those things that are on the shelf or in the lab right now, it's completely irrelevant. It's distraction. The whole enterprise is about selling Roundup. And that little molecule over there that you see on the right side, it's glycine methylphosphonate. It's a very small molecule, and I will talk about it in details later in the talk, because it's actually very important, and it's actually incredibly toxic. There is no doubt that the gene revolution, that GMOs have been incredibly successful because it is about weed management. And as you may or may not know, weed management for the farmers is really the #1 consideration. What kind of chemical to use, when to use them, how you mix them, et cetera, without avoiding damage to your crop. And here comes this technology 18 years ago that tells the farmers to completely ignore your weeds. You don't need to think about your weeds anymore. They're completely irrelevant. Just ignore them. Plant your seeds. And when your seeds come up a few inches, then you spray. And this is completely revolutionary. You don't normally spray a herbicide on your crop. You're going to kill your crop. But your seeds have been engineered to tolerate the herbicide. And to the farmers, it's a miracle. It's magic. It's wonderful. They were promised, of course, that you just spray once, and then all your weeds have disappeared by magic, which is wonderful. There is no doubt that the technology has been incredibly successful. 90% of corn, 90% of soybean, over 90% of soybean, 100% of sugar beet, canola today are engineered, because the farmers absolutely love the technology. It makes their life incredibly simple. So how are they created? Well, it's not too difficult to imagine. This is a gene gun. The plants are engineered using what looks like a hairdryer. It's a little handgun, and you're shooting micro pellets, very tiny pellets of metals, on which you have coated the piece of DNA that you want to introduce into the plant. Now, the DNA is just a code. It's the code for the plant, how to make the protein. So you just go to the library of a bacterial library, actually, they're all bacterial genes, and you take the code you want, and you put it on the micropellets, and then you shoot. And some of the pellets find their way into the nucleus of the plant cell, and somehow, by a process that we completely ignore, we have no idea how it happens, but somehow the code, the piece of DNA, will move into and be integrated into one of the chromosomes of the plant completely at random. We have no idea how or why. So this particular, and then when you've done, then of course you disintegrate the tissue, you have single cells, you put them on a petri dish, you don't know, it's a very inefficient process, you don't know which cells have been successfully engineered, and so now you have to find out which cells have been engineered, and to do that, before you start, of course, with the gene of interest, you put another gene, a marker gene, which is normally, usually, most of the time, at least until now, it's an antibiotic resistance gene. As a bacterial gene, you know that bacteria can become antibiotic resistant. So it's easy to find an antibiotic resistance gene to put with the bacterial transgenes that you want to introduce into the plant. And so now you have petri dishes with a carpet of cells that were shot, and each cell has an antibiotic marker, antibiotic resistance gene. You put antibiotic into the petri dishes, and all the plant cells die, except for the ones that have been engineered, that now have the gene of interest, which is the gene that will make the plant resistant to the herbicide, and the antibiotic marker gene. Now, the gene of interest is called enol pyruvyl shikimate phosphate synthase, EPSPS. You don't have to remember that. But the thing that I want to emphasize here today is that, as it's pretty obvious, we're talking of direct gene transfer. And that's the expression. You can also call it horizontal gene transfer. Direct gene transfer. This is what bacteria do. Bacteria exchange genes directly with one another. There is no sex involved. When the bacteria evolved into your eukaryotic cells, which became the plants and the animals, they lost that ability to do direct gene transfer. You cannot give your genes to the person sitting next to you. You need sex. Plants and animals do sex. Vertical gene transfer. You get your genes from your parents. You don't do direct gene transfers. So what we're doing here is a technology that is really borrowing from what the bacteria are doing normally, and we're doing that to the plants or the animals. And there are consequences to that. There are actually severe consequences to that, and I will come to that. When the gene of interest, the EPSPS gene, has been integrated into one chromosome of the plant and the cell has been engineered, then with tissue culture technique, you just regenerate a whole plant. And when you have a whole plant, well, you multiply it and you can get 500 million acres of it, because this is how much we have today. 500 million acres of genetically engineered crops on the planet. And you can tell the little symbol on the right side, Roundup Ready. So this is the name for those crops. They are Roundup Ready. They are now tolerant to the herbicide. And that's what GMOs are. 90% of 500 million acres are engineered to tolerate the herbicide. Everything else is distraction. So what do they deliver? Well, the biotech industry is very quick to tell us that the whole thing is about to reduce the pesticide. And I'm really fond of saying this. You know, imagine a large chemical corporation, and a group of engineers from that corporation comes to the board of directors to make a presentation saying, we have this amazing technology. We are going to reduce our sales by 50%. So, of course, this is not what has happened at all. In the last 18 years, the sales, the volume of Roundup has been increased by 1,500%. So 18 years ago, it was a minor herbicide. It was successful, but it was really a minor one. And of course, today, it is the most popular pesticide ever on the planet. No reduction in the use of pesticide, of course. Not only that, but in the last 10 to 12 years, the plants have, the weeds, the plants have adapted, the plants have evolved as any normal organism would do. Anybody, any scientist, any biologist would have predicted that, and they did, that the bugs or the plants would adapt, and they did. So now we have weeds that are very, very resistant to the herbicide, which means that in the last 10 years, the farmers have sprayed, and then they have sprayed again, and then they spray more, and then they increase the rate, the concentration of their spray, because the plants, the weeds are not dying. So much so that today, more than half of the acreage of the USA is infested with one or more of 40 species of resistant weeds. We call them superweeds. There is no solution. The only solution there is what I call the chemical treadmill. The only solution is that the chemical company has come up with the next generation. So now we are going to spray 2,4-D on the whole continent. And 2,4-D has a history of toxicity. Some of you may not have been born before 1945. I was born in 1946, so I'm not one of them. But you do remember the Vietnam War, and that was Agent Orange. No reduction in the use of pesticide, of course, quite the opposite. But really, we do need this technology to feed the world, don't we? Because this technology increases yield immensely, and it's a wonderful thing. Well, think about it. I just explained to you, you just take a piece of code that will make the plant tolerant to the herbicide. Who talked about yield? There's no yield. Yield is a very complex genetic trait. If you want to increase yield, you need to play with hundreds of genes. You cannot do that with this technology. This technology is really good at inserting one gene, perhaps two, or even a handful of genes into the plant. But you cannot, absolutely cannot, touch yield or salt tolerance or drought resistance or any other complex genetic traits such as those. There is no increase in yield. Actually, it's quite the opposite. There are several studies done early on, 10, 12 years ago, by universities in the USA that did show that there is a yield drag, 5 to 10% less yield. And it's just because the plants that have been engineered have been damaged. The genome has been damaged. I call it collateral damage, and I'll come to that. There is no increase in yield, period. You may get good weed management, or you may get good pest control, and that's a different topic, but you do not get increased yield because of this technology. And this technology absolutely is innocuous. It does not impact the environment at all. Well, you've just heard about genetic pollution. This is what I call it. Genetic pollution. And we've just heard about Mr. Steve March, who is right now battling the corporation because he has lost his organic certification because of genetic pollution. This is contamination. This is cross-pollination, as it should be called. There is no way that you can prevent pollen from flying. Engineered pollen will fly all over. Every crop is cross-contaminated. So this is a serious problem for organic growers. And now that the technology has been so successful, there is such a thing called the right to farm. And if you're an organic grower and you want to complain or you want to sue, you will be against the right to farm because because the technology is so incredibly successful and the farmers have adopted it in drove, they have the right to farm and they have the right to contaminate your crops. And I'm not going to take the time today to talk about other forms of genetic pollution which are just as, or perhaps even more important. There are some serious concerns about spreading antibiotic resistance. Every cell of every plant of 500 million acres contains one or several copies of an antibiotic resistance gene. And if you do the math, you will come with 10 to the power of 22 or 24. That's a lot of antibiotic resistance gene. And there are several papers that have shown that it's practically not possible that the bacteria in the soil could pick up bacterial transgenes from the plants that are decaying in the ground. And it's very difficult to show, and my opinion is that with 10 to the 22 or 10 to the 24 antibiotic resistance gene loose in the environment, it is happening thousands of times every day. But we won't discuss that. There's also another form of genetic pollution that you might want to hear about, is what happens in your guts. Because you do know that you have hundreds of billions of bacteria in your guts. And they will pick up bacterial transgenes from the food, from the plants that you have. Because that is well known, that by the time your food is in your lower intestine, it has not been quite fully digested. And there's definitely studies showing that the transgenes can be picked up by bacteria in your guts. And I'm not concerned about the gene for tolerance to Roundup. I'm more concerned about antibiotic resistance. So yes, huge impact on the environment. But we're all here to discuss safety and they're safe to eat. I mean, basically, Monsanto will tell you that their crops are not just safe to eat, they're actually more nutritious. And that comes from a concept that was put forward in 1993 by the OECD. It's called substantial equivalence. And substantial equivalence really means that, yes, the plants have an extra gene or two, or a few more, but they do look like normal plants. I mean, it's corn, and it looks like corn, and it tastes like corn. So much so that they are equivalent. And really, the way it works is that, you know, a chemical company or any company that has created a new variety of corn or soybean or canola, or they want it protected. So they need a patent. They go to the patent office, and the patent office says, well, if you want a patent, you have to tell us and show us and prove that you have something completely new and unique. And the chemical company says, I have this new variety of corn, and it has a new gene and a new protein, and it's completely new and it's completely unique. There is nothing like it on earth. And they get a patent. And then the next day they go to the regulatory agencies and they say, well, it's corn. And it looks like corn, it tastes like corn, it grows like corn, and it's just like all the other varieties of corn. It's completely substantially equivalent. And they get registration to commercialize their crop. That's substantial equivalence. It's a bit of a magic trick. And we do know as scientists, and this is published many, many times, many studies have shown that, I talked about collateral damage. When you insert a new gene at random in the genome of a plant, you have absolutely no idea where it's going or how it's going, and you are interfering with the regulatory sequences in the genome. This is shown. This is known, this is published. So much so that there are basically, you are really interfering with the code, and you are creating new proteins. Some genes are damaged, some genes are influenced, some genes are, the regulatory sequences don't work well, and you are creating new proteins. And There are many studies showing this. I hope you can read what's on the screen. In this particular study, I'm going to show you a few scientific studies. This is all. I'm putting the scientific references at the bottom. In bold is the name of the journal, the scientific journal, the title, the date, etc. And I'm summarizing the findings on top here. 43 proteins in engineered corn plants were significantly disrupted compared to non-engineered plants. Another study showing alterations in many proteins in tomato, transformed with a particular gene. And all this confirms a prediction. In 1996, 1996 was the first year, was the year when the first crops were registered in the USA, corn and soy. The scientists of the Food and Drug Administration in the US all talked to each other, and they had a broad consensus, and they went to talk to their director to tell him that this kind of crops should be thoroughly investigated and tested before they were commercialized, because they knew that engineered crops could contain new proteins, variant proteins, rogue proteins, altered proteins, mutated proteins, truncated, and those, some of them at least, could be toxic. This is 1996. The director of the FDA ignored them and registered the crops. And perhaps you know his name. He had been a lawyer from Monsanto. He was put in place as the head of the FDA in 1996 to register the crops, and soon after that he became a vice president of Monsanto. So in 1999, the first alarm bell rang very loud. This is a piece of research that was published, that was done at the Royal Institute in Scotland. The European Union was not going to take the word. I mean, they knew. They knew as much as we do. They knew what was going on, and so they asked one of their top scientists, one of their top toxicologists, to put together a protocol of research that could test the engineered crops to make sure that they were not getting any false positive or false negative, and that's what he did. He worked with engineered potato, and he showed that indeed after 3 to 4 months of feeding engineered potatoes to his rats, there was something toxic in the engineered plants. He went public. He was fired for it. He went public before he was before he published. He was fired for that, or I mean, there was a big political scandal, etc. But his research was published a year later in The Lancet. The Lancet is perhaps the most prestigious medical journal in the world. And he had the electron micrographs to show the damage. He called the damage to the lining of the stomach and the intestine, he called it precancerous. This is 2003, mice fed engineered soy have damaged liver, pancreas, and testes. This is 2012 in the US. Rats fed GE corn have damaged liver, kidneys, testes, and small intestine. So basically, the digestive tract is hit first in the rats. And this is a study done by Monsanto, a Monsanto group of scientists who fed engineered corn, Roundup Ready corn, to the rats for 90 days, exactly 90 days. And they found nothing. The rats, after 90 days, were very happy. There were no differences anywhere. The statistical analysis showed that there was absolutely no effect. And the conclusion was that this study complements extensive animal feeding studies with Roundup Ready corn, confirming it is safe and nutritious. That was published in 2004. And one of the scientists, that particular study was done because the European Union asked for it. And one of the scientists in the European Union in France was really intrigued because he had already done some preliminary work and he found quite different results. And so he asked to see the results of the study, but as you know, it is all patented and secret, so it was refused. He went to court, and after some legal wrangles, was allowed to see the results of the study, all the numbers, and he decided to redo the statistical analysis because somehow it hadn't been done properly, and he showed that after 90 days there were some signs of preliminary toxicities to several organs, notably the liver and the kidneys. He showed this statistical analysis to the regulatory agencies in France, and he was declined. The regulatory agencies called his study, his statistical analysis, biologically irrelevant. The significant differences that he found were biologically irrelevant. And he wasn't happy with that, so he decided to repeat the Monsanto study. Exactly the same. He repeated the study, he used exactly the same strain of rats, he used the same protocol of research, but he decided to increase the length of the study from 90 days to the whole length of the rats, the whole length of time of the life of the rats, 2 years. And because every engineered plant is sprayed with Roundup, and he had done some preliminary work already with Roundup showing that it is toxic, he decided to have some tests of Roundup as well included in the study. And he published his results in 2012. And what he found, of course, that was after 90 days, exactly as in the Monsanto studies. I cannot speak about this study because it does not exist. It has been retracted 3 months ago, but I'll come to that again. Exactly as in the Monsanto study, after 90 days there were preliminary signs of toxicity, and after 4 months there were very clear signs of toxicity to the kidney and the liver. And of course, as time went by, the rats became very ill, and after a few more months, some of them died and many of them developed tumors, breast cancer. And he published his results in 2012 in a peer-reviewed journal, of course. And you would not believe the amount of noise that was raised by the biotech corporate science community. These are not toxicology people, these are biotechnologists. They don't know a thing about toxicology, but they definitely know when something is not right. And so they criticized the study extremely heavily, very loudly. They wrote letters to the editors, they wrote letters in the newspaper, opinion letters, etc., etc. And the reviewers and the editors of the journal decided to hold their ground, and no, they did not retract the study. The study had been done properly. Oh, some of the critiques were about the strain of rats. The rats had developed cancer. Well, no wonder he had used a strain of rats that developed, that is very prone to cancer. It's called the Sprague-Daly rat, strain of rat. But this is exactly the way. It's a very common strain of rat to use in toxicology studies. It's used most of the time, and that's the one that Monsanto had used. And he also had done all kinds of things. He didn't know what he was doing. He didn't know how to do a statistical analysis, etc., etc., etc. And where things are becoming really interesting is that a few months after the publication of the study, the publishing company put another additional biotech reviewer, or editor, should I say, on the editorial board of the journal. somebody who had links, he had worked for Monsanto for many years, so with an obvious conflict of interest. And it didn't take very long before there was a review of the study, and it was retracted 3 months ago. This study does not exist. Nothing happened. Well, Roundup is really half of the story. Roundup is the herbicide. Roundup is sprayed on all those engineered crops, meaning that the food that is made with those engineered crops, by definition, contains residues of the herbicide. And this herbicide, when I was in graduate school in the 70s, when it started its career, this herbicide was one of the most non-toxic product ever. It was, you spray it and it disappears in the ground within days. It has no acute toxicity. It's like water. And today, this is what the farmers are told. It is like water. You can drink it. Glycine Methylphosphonate. This molecule was invented in 1964 by a medium-sized chemical company in the USA as a descaling agent, as a chelating agent. A chelator is a molecule, usually a small molecule, and all the derivatives of phosphonic acid are chelating agent. A chelator is a molecule that will grab onto metal ions. That's what it's designed for. A chelator is a wonderful product if you want to clean up soil or water of toxic metals or industrial products of toxic metals. It's great. But it's not that good in your food. A chelator will compete in your cells, in your body, for metals, iron, calcium, cobalt, manganese, magnesium. Glyphosate, which is the active ingredient in Roundup, glyphosate is a particularly broad spectrum and very strong chelating agent, a very successful molecule. And in 1970, so imagine you go to your doctor and your doctor says you're very pale, you're anemic, I'm going to do some blood work. You come back to your doctor 2 weeks later, yeah, you're anemic, I'm going to give you iron pills. Because in your blood you have a molecule called hemoglobin, and in the center of that protein there is an iron atom. The protein does not work without. Imagine that next to your molecule of hemoglobin you have a tiny molecule which steals the iron. Your hemoglobin does not work anymore. It cannot work with the metal ion. Glyphosate is incredibly good at competing for all kinds of metals and basically preventing many enzymes from doing their work. Disabling proteins, enzymatic proteins, that's what it does. That's where the toxicity lies, nothing else. A chelating agent. And because it is very good at what it does, at preventing proteins from working, not just in plants, it basically works on every living cell, including bacteria, so much so that glyphosate is patented as an antibiotic in 1970. So glyphosate was patented as a chelator in 1964, as a herbicide, and it has become, of course, extremely successful, the most successful pesticide ever. In 1970, and that's when Monsanto made history with it, and in 2010 they were bold enough to patent it as an antibiotic. We know that antibiotics are not a good idea in your food. We have many studies showing that it will be toxic to the microbiome of poultry, of cattle and pigs. I hope you all know what the microbiome is. The microbiome is an organ that every animal owns, has. And the microbiome in your own body is an organ that is just as important as your liver, your kidney, or your heart, and perhaps your brain. The microbiome is the hundreds of billions of bacteria that you have in your lower intestine. And if you think they just sit there doing nothing or perhaps feeding at your expense, that's not the case. You are a symbiotic organism. You are not a human being. You are vastly outnumbered. 90% of your cells are not human cells. 99% of the DNA in your body is not human. It is in your microbiome. These bacteria, and there are thousands of species of bacteria in your intestine, these bacteria are absolutely essential to your life. They are in charge of your immune system. They are in charge of your brain functions and happiness. They make 90% of the serotonin that you have in your brain. There is more connection, nerve connection, between the microbiome and your brain than to any other organ. This is how important this organ is. So, feeding your microbiome a daily diet of antibiotic is really not a good idea. You will get sick. Not necessarily very quickly, but you will. As happens other animals. We have no studies on humans. So this is mostly for women, of course. Glyphosate induces human breast cancer cells. Food and Chemical Toxicology, 2013. And this is for the boys. Roundup disrupts male reproductive functions, also in 2013. And we have known for a long time now that glyphosate inhibits cytochrome P450. Now, you probably don't know what that is, but cytochrome P450 is a family of proteins, of enzymes, that are absolutely essential to many, many services in all our cells. And these family of enzymes are inhibited or impaired by this chelating agent. If you impair these family of enzymes, you can expect these symptoms: ADHD, bipolar, schizophrenia, depression, autism, Alzheimer. And we know already that, in rats and mice, we have seen those symptoms: gastrointestinal disorders, kidney failure, liver damage, infertility, and diabetes. And when I say gastrointestinal disorders, this is not, tummy ache after too much of a meal. I'm talking celiac and Crohn. Very, very important stuff. The biotech industry has a really nice quote. Millions of people have eaten trillions of meals with engineered food. Thank you, Anne. And there is not a single report of illnesses. You've probably heard it, you've probably read it, and if you say it 1,000,000 times, it becomes true. This is the most vacuous, empty statement. There is no follow-up. There is no epidemiological studies. There is absolutely no way of knowing who is eating what. And that's the status today. We have no idea of who is eating what and what it does. And the biotech industry will tell you that it's all very safe, and not a single report of illnesses has ever been published. All we have is statistics from the US Department of Agriculture that publishes the amount, the volume of glyphosate, of Roundup, that is sprayed on crops every year. in the USA. And we also have statistics from the US Center for Disease Control in Atlanta, Georgia, because they keep records of who is hospitalized for this particular injury or damage or disease. And you can go through these databases, and you can put things together, you can put pictures together from that. And this is what you can see if you do that. Oh, by the way, I did not tell you that Roundup, the grains are not engineered. Wheat is not engineered yet. Well, it is engineered, but it hasn't been commercialized. But I learned a few weeks ago that grain is sprayed with Roundup a few days before harvest. That's normal practice, because it makes it much easier to harvest. The plants die, the grains dry fast, and it's much easier to harvest. And since Roundup is like water, there is no problem. So grains are not engineered, but they are definitely laced with Roundup. So this is looking at the databases of the US Department of Agriculture and the US Center for Disease Control. This is the volume, and the lines are the volume of Roundup sprayed on corn and soy here, and this is diabetes symptoms. And you can see that starting in the mid-1990s, the curve goes up very fast. Yeah, thank you. Autism. Now, autism is a very strange disease. Autism, 20 years ago, about 1 in 10,000 kids had autism, was on the autism spectrum. 1 in 10,000. 20 years later, today, 1 in 50. 1 in 50 children is on the autism spectrum. And this is what the curves look like, starting in the mid-1990s, when the engineered crops took off. Intestinal infections, and this is the number of deaths. People dying from intestinal infection because the bacteria in their guts really just gave up. Again, starting from the mid-1990s. This is celiac in the US. Again, climbing up, starting in the mid-1990s, and this is the volume of Roundup sprayed on wheat. This is in Canada. This is the only curve I could find in Canada. This is in Alberta, at Children's Hospital in Alberta, again, climbing up, starting in 1996, when GMO canola took off. Kidney damage. I could show you 20 of those, but I think you get the picture. The rest of the world is not stupid. No pun intended, nothing personal. But you know, the governments of the rest of the world and the people and the scientists of the rest of the world, I mean, can read between the lines. They can read the scientific literature. Yes, there is at least 100 published studies from Monsanto or the biotech corporate science community that will show you with statistics that it's perfectly safe and there is absolutely nothing to fear, and anybody that tells you the opposite, like me, is either a quack or an activist. 64 countries today have decided either to ban or to regulate, to label GMOs, and somehow the biotech industry is really not very successful in most of the rest of the world. But in Canada and the USA, we are really, really in the dark. I call it the biotech bubble. So very quickly, this is what the rest of the world see, that the crops are sprayed with a chelating agent which impairs many enzymes. It's antibiotic. There are toxins, toxic proteins from the genetic engineering process, of course the health problem, and then of course there is the genetic pollution problem. And I was speaking 2 days ago to a group of kids in Ajax, and this is what I told them. Because this is pretty dark, you know, what can you do? I mean, you go to the grocery store And if you watch, there's a new video by Bill Nye, the Science Guy. I was talking to the kids. Bill Nye, the Science Guy, I watched it a few days ago, telling you for half an hour that this is perfectly okay and it's completely safe, and we've been doing this for 10,000 years, etc., etc. So this is a really dark picture, because you go to the grocery store and the vegetables and the fruits are not engineered, except for one or two exceptions. But everything else, the meat, the dairy, the prepared food, the junk food, the canned food, the baked food, 80% of that contains canola, soy, corn, and sugar. And all that is laced with residues of Roundup. The legal residues of Roundup from 1994 up to 2013 was 0.2 parts per million in the food. And in 2013, the EPA, in their wisdom, decided to increase that limit times 100. 110,000% increased in legal residues of Roundup in the food. I don't know where the data came that showed that it was perfectly okay to do that. There's no studies, but that's the reality. Anyway, talking to the kids, I told them to do, so this is the picture, I told them to do this. Turn off your TV. Makes you stupid. It does. Increase your food budget, and that I would tell everybody. Increase your food budget by 50%. I don't care how poor you are. Just shave off everything else. It's food, it's your body, it's your biome, it's your microbiome, for God's sake. Take care of yourself. Make your body your temple. Increase your food budget. Go organic. I know you would approve. Learn to cook. And I said, learn to cook. It is proven scientifically that if you learn to cook, you get more dates. And then I go empower yourself and, talk to your neighbors, talk to your grocery store manager, march, etc., etc. Thank you very much. Oh. If you don't know this publication, I highly recommend it. You don't need a university education to read it. It's a compilation of studies, peer-reviewed studies, put together by a genetic engineer in London, England, and written by a science writer, so it's written in perfect English. And it's easy. You can download it from earthopensource.org, and it's free. 100 pages, and it's a really good read. Thank you.